So regarding the pathogen, this picture that you observe here, it was produced by us in collaboration with a company called Eye of Science. What you observe here is Fusarium, a mycelium of Fusarium just going out from the pseudostem of a plant that I inoculated at my lab. So talking about the pathogen, as you know better than me, Fusarium, very important group of fungi, more than 1,000 species. The pathogen, the Fusarium from banana had been related to the Fusarium oxysporum complex for many years, which includes endophytes, saprophytes, and also pathogens, not only to plants, but also animals. Some of them produce toxins in cereals. And in plants, they cause wilting. They have also these forma specialis, which makes them very specific to one host. Like, for example, the cubense is specific to bananas, the pizzi specific to peas, and like this, there are many, many more. The Fusarium from banana causes the Fusarium wilt or Panama disease. For many years, it's been known as Fusarium oxysporum forma special cubense. However, there is a recent and revised taxonomy of the fungus by Mariani, one of my colleagues, that I would really recommend you to read. And you can see great pictures of Fusarium and morphology and from different locations. Fusarium produces this type of conidia, macroconidias, and chlamydospores. Especially the last ones are very persistent in the soil, and there are indices, there are ideas that it can be even decades. So of course, this need to be proved, because the only reason why people say that it can be in decades, because when they said, for example, in the Philippines, there are areas where for 30 years there were not bananas. And when they put a Gros Michel there, again, the Gros Michel is sick. So they think that the Fusarium was just waiting there. So there are many theories. One is that they were really just latent in the soil, waiting there. But the most close to reality theory is that they were just in other hosts, what has been already proved by my colleagues. So these of the decades, it has to be proved, but still it can stay there for many years in different hosts. There is not an efficient control. So Fusarium is very weak actually in the lab. If you apply fungicides, you can kill it. But the challenge is when you want to apply this in real conditions in the field, because one of my colleagues also started an experiment in which she measured different areas of the soil in deep, and she found Fusarium even in 1 meter deep. The most scary thing is that it spreads extremely easy in 3 ways: by plant material, by soil, and by water. So, and among these 3, you can imagine all the possibilities. Soil, for example, in the shoes, tools, animals, containers, all these kind of things. Water when it's raining, for example, because when you have these plantations on bananas, you have to build this drainage, and all the water goes there and moves around the farm. So that's a way how it moves. Also, there is an example in the Philippines in which my colleague took samples from the river, and she could isolate Fusarium from the river. If you like this part of the taxonomy, I have some cartoons in which I explain the taxonomy of the Fusarium through the years. Something I want to say here is about this one, in which the Dr. Brandes was the first person that demonstrated the pathogenicity of Fusarium cubense in that time, when he inoculated plants with contaminated soil. It's interesting because 100 years ago, we still do the same to prove that the strains are or not pathogenic. So that's how we do it. The life cycle, again, is very similar to other soil-borne pathogens. There are spores, macroconidias, but essentially chlamydospores that are in the soil. They germinate, They grow through the cortex, reach the epidermis, and then from there start to grow and is easily moved through the rest of the plant thanks to the xylem. And then what it does is just grow inside, blocking the vessels, causing this collapse of the plant. So at the beginning, after a few hours of contact with the pathogens, you can see here, some of the spores are already attached to this. This is a tip of a root, and these are the macro and microconidias. What happened then is that the fungus start to grow, not only in the vessels, as you can see here and here, for example, all this white is the Fusarium, but also these areas that are empty areas in the pseudostem. This is a pseudostem of the banana. And this picture that I showed you at the beginning is a magnification of one of these areas. But not only the empty areas. It's been proved now that the Fusarium also pierces and crosses the cell wall, as you can see here in many of these are the cells, and Fusarium is just crossing